right, so I want to thank everyone for for being here. I'm coming from the medical school at Rutgers University, and I want to assure you that I actually have a sincere interest in the origins of life, and I didn't just want a free trip to Japan. And I've actually, as part of the medical school, I don't know how to operate a computer. Let's see. There we go. So as part of the medical school, one of the things that I do is develop drugs. And we're very interested in actually using structural modeling of proteins as an approach to designing new therapeutic molecules. But at the same time, in my heart of hearts, my fundamental interest is in origins of life and how proteins fold and how they evolve. And one of the things that we've been interested in, and I've been studying now for over 15 years, is the emergence of homochirality. And I want to thank Professor Whitesides for introducing this idea of the Leventhal's paradox, because for me, protein folding is an important obstacle for biomolecules to overcome in origins. And so one of the things that we were studying is, using our computer simulations, why are proteins homochiral? And what we find is that when sequences are homochiral, they're able to fold more quickly. They're able to find their folded state in a much smaller phase space. This is the phase space, for example, of a homochiral sequence, whereas if you're heterochiral, you have to sample a lot more conformations in order to find a unique state. And so for us, that was very interesting, because with a very simple simulation, we could see some very fundamental properties of molecules. And then once we realized what were sort of the guiding forces that were driving homochirality, we were then able to use that to sort of violate those principles and start to design small peptides. For example, this one. This is a peptide made out of all L-amino acids except for the N and C termini of the peptide, where there are D-amino acids that cap the ends of the peptides and hold them in a folded state. And this peptide, even though it's only 8 amino acids long, is very stable. It's able to find its native state very quickly and very efficiently, and then block an interaction that's key in the life cycle of the influenza virus. And we're now developing this as a potential therapeutic peptide. So even though I'm at the medical school and I'm studying origins of life, there really is a nice connection between these two sides of my life. And so when I first came to the medical school about 11 or 12 years ago, I was very excited to meet Paul Falkowski, because he got me engaged in another origins of life problem, different from homochirality. And so this is the project I'm going to talk to you about today. It's primarily the work of a postdoc in the laboratory, Hagai. Hagai came to us from a microbial physiology lab in Israel, and he very quickly became a structural biologist, so I'm very proud of the work that he's done. And this is also part of a larger team: Douglas Pike, a valuable graduate student in the lab, postdocs Eli Moore and Stefan Sen, and then also collaborations with Jana Bromberg, who you've heard about some of her work earlier, and then support from a number of foundations and associations. So Paul, when I first started talking to him about this, essentially was very interested in the origins of these complex protein nanomachines, these oxidoreductases that were capable of doing very efficiently sophisticated coupled electron transfer catalysis. And we call these proteins nanomachines because they are massive macromolecular complexes. And these are critical reactions, and at some point, they must have emerged because they were critical for life's processes. But we couldn't imagine something like a nitrogenase climbing out of the primordial soup on its own. There had to be some kind of a simpler ancestor. And we're really interested in what these ancestors look like. And maybe as you walk back, there may even have been small peptides in the early Archean or late Hadean that had similar function that sort of themselves emerged from the primordial soup. But as we all know, the problem is we really don't have any information about what these molecules look like. There are no molecular fossils for what happened at these early stages. All we have are the extant molecules, and then some geological information, some mineralogical information about what the rocks may have looked like at that time. So the question is, how can we sort of walk back the common ancestors of these modern extant complex proteins and figure out what these original molecules may have looked like? So I call these things massive nanomachines because when you look at some of these enzymes, they really are incredibly complex, right? So these are, they're doing critical reactions that, as we've heard from many of the talks throughout the session, they take advantage of this extant disequilibrium on the planet, and it's a great source of energy. But you can't imagine, a protein like this emerging spontaneously. And again, this is a manifestation of the Leventhal's paradox. So, think about something much simpler, a protein that's maybe more medically relevant. This is the beta chain of insulin. It's only about 30 amino acids, right? And so we know that for a protein to fold, it has to go from this unfolded state into a single native state. And each amino acid in this chain has 2 rotatable bonds. And if you think about sort of the staggered eclipse conformations of bonds, you could minimally have 10 conformations per amino acid. So you're talking about a total of, combinatorically, a total of about 10 to the 30th power unfolded conformations, out of which it finds one folded state. So even for something as small as the beta peptide of insulin, this is a massive problem. If you think about the other sort of side of the Leventhal's paradox, now this is, how did that particular protein evolve? You can also see that this is a massive combinatorial problem. So even for something this small, how do you find a specific sequence that has this function from the 20 to the 30th power unique sequences, right? So even if the functional footprint of something that acts like a beta insulin peptide is, let's say there's 1 billion sequences that could do that, or 1 trillion sequences that could do that, you still have a huge phase space to search in order to find a sequence that's going to have this function. And this is something that's only 30 amino acids long. If you look at something like nitrogenase is about 2,500 amino acids. And so how does something this complex evolve when you have such a massive conformational space to search and such a massive sequence space? And the answer, this is not something that's new to oxidoreductases or new to this particular project. But we know that proteins really are assembled by much smaller domains. And so there's a couple of ways that nitrogenase solves this problem. One is that there are multiple proteins that associate to form the macromolecular complex. There's symmetry that a lot of proteins take advantage of, so you can double or triple or multiply your complexity just by taking advantage of symmetric transformations. And then the problem becomes a lot simpler. Now you think about, how did these individual modules evolve? And the phase space that they have to search is significantly smaller. So I say that the solution is, let's identify what these individual building blocks are, and they would be a much simpler thing to imagine evolving. But that's not a very easy thing to do, right? We can't just sort of carve out pieces of nitrogenase and say that this piece evolved and then this piece evolved, and these are our modules. This identification of these ancient building blocks is itself a very difficult problem. And then also, once you identify what these domains are, what these modules are, I think another critical problem is figuring out, how did they self-assemble? How did they aggregate to make these more complex functional molecules? So the way that we approached this problem was to say, if you look at something like an oxidoreductase, so this is fumarate reductase, for example, let's ignore most of the protein and let's really just look at the important part of this protein, the one that's involved in electron transfer. And electron transfer is really just mediated by this chain of metals, this little necklace of metals that's running through the center of the protein core to its active site. And if we argue that these are really the functionally important parts of the protein, then we would imagine that the fundamental modules that assemble into these larger molecules must be centered around these metals. So essentially what we did was we went into a database called the Protein Data Bank. And for those of you who are not familiar with this data set, the PDB is a repository for the high resolution structures of proteins. So these are atomic resolution structures of proteins, not just oxidoreductases, but all kinds of different proteins, hemoglobin and so forth. And there's over 100,000 different proteins that have been deposited in the PDB. Which now I think closer to about 150,000. And of these, about 10,000 or so have metal centers in them. And what we did was we essentially took all of those proteins and we looked at what we call a microenvironment, which is essentially just the amino acids that are within a certain distance of the metal center, which we think is the important part the protein for electron transfer. And we just excavated all of these out of these proteins. So we had about 30,000 microenvironments. And then we tried to classify these into a smaller set of modules based on their metal type, but then also based on some sort of structural similarity. And for those of you who have done comparative structural analysis of proteins, you know that this is not a trivial thing to do, particularly when you're looking at alignments that are of sort of intermediate quality. So for example, what I'm showing you here, these are two modules that are centered around an iron-sulfur cluster. These are about 15 angstroms in radius, so we're essentially looking at all the amino acids that are within 15 angstroms of that metal center. And we want to ask whether they have similar protein structure holding the metal in place. And these are two examples of the types of alignments we could get. And on this axis right here, we have a similarity score. This is essentially telling us how well do the atoms of those two environments align. And you can see here in this alignment right here, the red and orange parts, these are the parts that align. You can really see only a little bit of the structure aligns right here. And then here's another alignment between two modules. And you can see, again, only maybe about 20–30% of those two structures align. And so they have about the same similarity score. And so if we were just doing standard structural alignment tools, we would not be able to say which one is a good alignment, which one is a bad alignment. They're both sort of on the edge of being an acceptable alignment. But what we know is that in order for these domains to function, they must contain this metal center, and this metal center is really sort of the functional nucleus of these modules. So really those metal centers must also align. So we're using the metal center essentially as a fiducial marker to tell us how good our alignments are. So even though these have very similar scores, here the metals are right on top of each other and we believe this alignment. And here they're far apart from each other and we don't. So this was a real breakthrough for us because it allowed us then to do this structure-structure comparison on a large scale and not have to go through and analyze each one manually and figure out whether we believe the alignment or not. And so we did this for all of these 30,000 modules, and what we found is that there were about, let's say, 1,000 different modules, and we could cluster all of these into these different classes. And that number 1,000 is not an exact number, depending on what your threshold is for similarity. You can make it larger, you can make it smaller. But we do find that there are a couple of domains that are a couple of modules that have a lot of members. And within these, we have the ferredoxin, which is not surprising, the cytochrome C, but then also a copper-binding plastocyanin, and then a 4-helix bundle that could either bind 1 or 2 metal ions in the center. So it looks like we have, for example, we have here a couple of Legos that are commonly used in metalloproteins. Now, what we're looking at here, this is essentially, the way that we're defining these microenvironments is the metal in the center, and then we're just sort of carving out amino acids that are within 15 angstroms of that metal. And sometimes this is a discontinuous piece of the protein. You may have loops that are going out to another domain, or maybe this part of the protein is coming from one chain, and this part of the protein is coming from another part of the chain. So why would you believe that this is actually a relevant module for evolution? So one of the things that we noticed was that when we look at the size distribution of these modules and we put them on a log-log plot, we see that they have a semi-linear relationship. And that is consistent with a model of domain evolution where you have duplication of these modules. So you can imagine that you have old modules that have been around for a long time and they've had a long time to duplicate within genomes, and so there's a lot of copies of those. And then you have, at the same time, innovation. You have new domains that are being invented, and those exist at a much smaller fraction. And so when you have this sort of a process of domain innovation and then duplication, you get this sort of linear relationship of module size versus frequency on a log-log plot. So this gave us some confidence that even though we are sort of creating these shaved pieces of proteins, that this was a functionally relevant and evolutionarily selectable domain that we could then think about in terms of its functional consequences. So now the question is, some of these domains, for example, like cytochrome C, contain hundreds or close to 1,000 different microenvironments extracted from the PDB. Am I saying that all of these modules have a common origin? They all came from an cytochrome C type domain, and now we're seeing them occurring in all these different proteins. Well, that's pretty hard to believe. So for example, here we're looking at just one of these sets of modules. So each of these dots represents one microenvironment from a specific protein, and an edge represents two microenvironments that have an acceptable alignment to each other. And so for example, to get from this microenvironment right here from one protein to this microenvironment right here from another protein, we would have to go through minimally 12 different intermediates to get from there to there. And we're not just staying within prokaryotes, we may be going in between to a eukaryotic module and then back to a prokaryotic module. So clearly these are not convincing evolutionary trajectories based on structural similarity alone. So another way of thinking about this, I think about, are we discriminating homology versus analogy? And really what I'm saying is that, for example, within a particular module like the ferredoxin or the cytochrome C, with similarity alone, we don't know if we're looking at all bird wings or whether there's bat wings and butterfly wings mixed into this data set. So that's an important thing to keep in mind, that structural similarity itself is not sufficient to prove homology. And I'm going to get back to this. Okay, so we have 1,000 or so modules that are being used to build these more complex proteins. Now, what's really interesting, if we want to understand the emergence of complexity within these systems, we want to see, are there any rules that can guide how these modules are connected together? Can we figure out the rules for how these Legos are assembled? And for this, we take advantage of the fact that we are really interested in electron transfer. We're interested in the ability of proteins to move electrons from one side of the protein to another, from an active site to a different part of the protein. And there, what we can take advantage of is that since we have the high resolution structures for all of these proteins, we know the distance between each of the metal cofactors. And there was a very important and influential study from Les Dutton's lab in the early 2000s where they looked at a set of oxidoreductases, and they looked at the distances between pairs of metal cofactors that were involved in electron transfer. And what you can see here is on this plot, here you have the distance on this axis between two metal sites within a protein in an electron transfer chain. And then on this axis right here, the log of the electron transfer rate. Right? And for all electron transfer chains within proteins, the metal cofactors are at most found by 14 angstroms away from each other. And at 14 angstroms, you're now thinking about electron transfer rates on the time scale of microseconds. Any further than that, then the electron transfer rates become too slow to really be biologically relevant. And so what we said was that, well, if we were interested in electron transfer chains, we can then just look for modules where the distance between cofactors falls within this distance cutoff. So really what we're doing now is we're going through the same data set of proteins, and now instead of connecting microenvironments based on structural similarity, we're connecting them based on their spatial adjacency within a protein. So we can say that, for example, this type of ferredoxin domain or this type of iron sulfur domain is often found next to a molybdenum site. This type of heme domain, for example, is often found next to an iron sulfur site. And we can build this map of spatial connectivity within oxidoreductases. So we did this, and this is what we got. So there are a lot of interesting things about this network. What I'm showing you here, each of these nodes now is not a specific protein site, it's a module. So it's the collection of microenvironments that all have structural similarity. The size of the node represents the number of connections it makes with other types of modules. So these edges are to other modules that are beyond our threshold for structural similarity. And then the edges themselves represent, the thickness of the edges represents the number of instances of a particular connection that we see. And we can see here that those same four modules that were highly represented in the data set, they also are, they make a large number of connections with other types of modules in this spatial adjacency network within the span. And so what rules can we get for the assembly of electron transport chains from looking at this? Well, one of the things that we noticed for about 30% of module-module connections, we have, instead of connecting one type of module to another, we have these loops. And so what a loop here represents essentially is a ferredoxin type module connected to another ferredoxin type module, or a cytochrome C connected to another cytochrome C, or a rubidoxin connected to another rubidoxin. And so, again, this is not something that is new to oxidoreductases. This is something that you classically see in a lot of different multi-domain proteins, is that the way that you make complexity or you make larger proteins from smaller domains is through duplication and diversification. So there are some very clear examples of this in oxidoreductases. You have these, for example, these multi-heme proteins in Geobacter, for example, that allow electron transfer from mineral substrates into the cell. Or here's an iron-sulfur wire that's made out of multiple ferredoxins that are connected together. We see similar things for plastocyanins, for ferritin, and so forth. But I think what's really interesting here is that it's not just sort of these very clear examples where you have these multi-cofactor chains, but nearly every module has examples of these sort of duplications. So clearly, an important rule for how do you build complexity is to just copy something and connect it to a domain of the same kind. So that's one rule that came out of this. But one of the other things that we found very interesting, and it jumps out at you if you color the nodes by the types of cofactors that they bind, is that all of the cofactors of the same type are connected to each other. So for example, here yellow represents iron-sulfur cofactors. And this is not just 4 iron, 4 sulfur. This is 2 iron, 2 sulfur, or something like rubidoxin, where you have a single iron and 4 cysteines coordinating it. So all of these are connected to each other. And a connection here, remember, does not mean structural similarity. So we're not saying that a Rieske type iron-sulfur cluster site looks a lot like a 4 iron, 4 sulfur from bacterial ferredoxin. They're structurally very distinct from each other. But what we're saying is that they're often found connected to each other. The same thing is true for these 4-helix bundle type single iron sites that are connected to a lot of other monometal binding sites. Same thing are true for heme binding sites. Same thing are true for copper binding sites. So this is actually very interesting. Why are we getting this metal segregation within this graph? And so there's a couple of explanations for this. So one would be that what we're seeing here, because these are essentially, we're arguing that these are electron transport pathways. So one argument would be that all iron-sulfur sites have similar redox potentials, and so the fact that you have all of these iron-sulfur sites connected to each other is essentially just a thermodynamic phenomenon, that this allows that you don't have any high barriers from transfer from one site to the next. But we know from protein engineering studies that you can have the same metal site and just make single amino acid changes around in the second shell around metal site, and you can move the redox potential by over a volt. So you can, for example, with iron-sulfur sites or with heme sites, have a huge tuning potential without changing the metal type. So it's a possible explanation, but it's not necessarily a very convincing one. Another one to think about is protein biosynthesis. So if you're making a protein that contains multiple cofactors, it might be easier in terms of assembly to have all the metal cofactors be the same, and then you can provide multiple iron-sulfur clusters to a single protein or multiple hemes to a single protein. But we know that there are many examples of oxidoreductases that have multiple different cofactor types in them. So those are two explanations, but another which we think is particularly tantalizing and one that we're now exploring experimentally within the lab is perhaps what this is suggesting is that these physical connections between modules represent duplication and then significant diversification. That you have, the iron-sulfur site constraining the first shell ligands, but then beyond that you get significant diversification of the second shell and the microenvironment around the protein. So in other words, that these connections in space may actually represent evolutionary connections. And this is, I would say that this is something that we still haven't proven, but it's the way I like to think about this is, we have this expression in English that if you're comparing apples and oranges, you're talking about two very different things, right? They're both fruits, but they're very different fruits from each other. One is citrus, the other is not. But what if you were to walk out and you find a tree that had both apples and oranges on the same tree? Now you'd start to say, well, maybe that expression doesn't make so much sense. Maybe apples and oranges are a lot more similar than we thought. And what we're thinking we might be seeing here in that span is what were originally thought to be apples and oranges occurring on the same tree. So if that's the case, then what we have now is a tool for discriminating analogy and homology. So if we go back, for example, to the heme binding cytochrome C module. So this was a module that had about 1,000 different members. And let's take this single module now and we cluster it using a Louvain clustering method. It's just one way of sort of classifying subgraphs within a larger graph. Let's say we classify this into like 8 smaller segments. And these connections here are based on structural similarity. And then we take that and we now generate a span for that. So now we say within those subgraphs, which ones are found spatially next to each other within the same protein. And what we find is actually within this larger module, there are subclasses, so that 1 and 2 are often found connected to each other, but we never see connections between 1 and 2 in any of the other cytochrome C-type modules between 3 and 8. So maybe there actually are 2 evolutionarily different cytochrome C-type modules that all they share really is just this chemical similarity that they bind hemes, but they have otherwise independent evolutionary origins. So in other words, what we're looking at here is convergent evolution of 1-2 class and the 3 through 8 class, but then these are all divergent from each other. So then that suggests if we look at go back and look at the network with this mindset that perhaps we have 4 or 5 or 6 fundamental modules. Maybe the ferredoxin obviously is a very tantalizing one. This may have been one of the first iron sulfur modules that then diversified into a number of other module types. The same thing with maybe one of these cytochrome C-type modules, the 4-helix bundle as a source, and then the plastocyanin for copper. So we're very excited about exploring now whether or not we can use these as sort of archetypes for understanding the evolution of these original oxidoreductase modules. So what I'd like to say at this point is that we can take something like a large complex oxidoreductase and decompose it into smaller modules. And we believe that these modules are behaving like evolutionarily selectable domains. They're functionally discrete, they're selectable. And we believe that this complexity likely evolved from the assembly of these smaller modules into these much larger complexes. And very simply through domain duplication to build wires, but then also maybe through diversification to develop these more functionally specialized branches of these electron transfer pathways. And perhaps we can start exploring these spatial adjacency relationships as a construction to look much more deeply into the fossil history of proteins, where we're not depending on the vagaries of structural alignments, which are themselves more sensitive to deep time than sequence alignments for making these relationships, but also using this as another way to establish connections between structural domains. And as with our work on homochirality and our ability to relate that to the design of therapeutic peptides, we also see in this case evolution and design as 2 sides of the same coin. So while we're studying the evolutionary relationships between these modules, we're also now thinking about ways of hooking these things up together to start to make nanoscale devices for bioelectronics. And Eric, you may look at this and you may see a bifurcating pathway right here. And we're actually very interested in going back to the span and think about, in addition to just pairwise interactions, maybe multi-body interactions between between these metal sites, is to look at the emergence of more complex topologies in electron transport. And so perhaps instead of having a single ancestor that has led to the emergence of all these different oxidoreductases, maybe we had several Lucas, or maybe you want to call it Lucas, that themselves assembled in different ways to make these modern extant nanomachines. And so what we're doing now is we're starting to ask, how can we walk from these very simple domains, which themselves are fairly functionally naive, to perhaps complexes where you have 2 or 3 domains that can do more interesting things. So we want to move in this direction towards the complex machines and see what are the minimal assemblies that give us the functional, the catalytic properties that we want. But also, what's interesting now is we already have fairly simple archetypes of what these original molecules may have looked like. Can we start to walk them back even further? Can we go backwards in evolution path before the common ancestor and start thinking about what these prebiotic peptides may have looked like? And of course, this is something that we heard a little bit about yesterday, thinking about how peptides may have interacted with minerals themselves that are already capable of redox catalysis, how peptides may have aided this. And I'll just show one slide that shows one of our forays into this area. We've been looking at bacterial ferredoxin for a long time. It's a protein that binds 2 4-iron 4-sulfur clusters. It's about 60 amino acids. It itself is clearly a domain duplication of about 20 to 30 amino acid domains. And what we've done is by looking at the reach at the business end of this molecule that is responsible for binding the orange sulfur cluster, we've been able to reduce this 60 amino acid protein about 5-fold to this small cyclic peptide, which is only 12 amino acids. And this 12 amino acid peptide is able to stably bind a 4-iron 4-sulfur cluster. This is an EPR spectrum showing that it has the classic signature of a 4-iron 4-sulfur protein. But what we find particularly exciting about this particular design is that if you notice here, if you look at the topology of this protein, all of the backbone amides, which are in blue, are pointing in towards the iron sulfur cluster. And this is something that a structural biologist, Milner and White, would call a cationic nest. So essentially all these backbone amides are creating a nice stable binding site for an iron sulfur cluster. And so what happens now is that because you have the stable binding site, we're able to take this complex and oxidize and reduce it thousands of times and it doesn't fall apart. So this thing has a redox potential close to that of ferredoxin, but it's very stable. And I've designed several iron sulfur proteins in my career as a protein designer, and the best that we've been able to do before this was about 16 cycles before the thing falls apart. And in fact, the most recent design before this fell apart after 1 cycle. So something that has this extent of stability is unprecedented. So we're very excited about this. And so designs like this that are inspired by these small domains may be a way for us to extrapolate back to what prebiotic peptides may have looked like. So I'll end there, and thanks again for the invitation and the chance to speak, and I welcome any questions.
Wonderful talk, thank you. And we'll take the first question from George.
That was really fabulous. I mean, extremely classy. A couple of quick questions. In these electron transfer chains, are you seeing hopping? Are you seeing drift? Are you seeing tunneling? What is the mechanism?
So we're agnostic to the mechanism. We don't know if we're seeing tunneling or if we're seeing hopping, right? I mean, you're looking essentially at connections between metal clusters that are within the context of the protein matrix. So if you're Harry Gray, then you would be looking for essentially hopping intermediates between these metal clusters, so aromatics, for example. And so one of the things that we're interested in looking at now that we sort of identified what these electron transport pathways are, see whether we see amino acids between them that may be affecting the conductivity, the beta of the environment that would help us delineate the mechanism.
Okay, and then one other question of the many I could ask. If you look at the genomic structure of these systems that have these hypothesized multiple domain replications, are they contiguous? Are they intron-exon mixtures? How do you actually get these piled together in the genome in such a fashion that you end up with the collection of contiguous amino acids that you see?
So we simply haven't done that, but I think that's an important next step, is to start thinking, because evolution doesn't happen in structure. You don't glom units together. It has to happen at the level of sequence. And previously we tried to look at this problem using sequence analysis alone, trying to extrapolate from one type of metal binding site to another through sequence intermediates. And I think that combining that analysis with the structural analysis would be a way to get at your question, definitely.
Hey, so great talk. For the amino acid sequences that are involved in these highly stable kind of small systems that stabilize these, this.
Yeah, sure.
Yeah, is there any relationship between the amino acids present in those and the biosynthetic pathways by which those amino acids are generated? Meaning, are they kind of potentially early amino acids, or are these something that came along quite a bit later?
I don't know. So I don't know if there's if we can only make these things with simple amino acids. What I'll say is that the sequence pattern that we're using here is very similar to the one that was proposed by Dayhoff and Vanek, which is that small 4 amino acid repeat. And critical to that is having a cysteine obviously binding the cluster, but then the flanking amino acids like lysine and glycine work very well for those.
Yeah.
Whoever has the cube,
May I?
the magic cube.
Yeah, hopefully quick. Stunning, like everybody else says. It's a question about the way you use this paradigm for evolutionary interpretation. It looks like this natural modularization gives you a kind of typology of functional states. And if I think about an evolutionary model, I want a model of states and transitions. If I think about what people often do in trying to recover old protein folds or old protein fold fragments, they look at the recruitment of a thing that's effectively a unit that can move, that can mutate, that can do whatever. Is it possible to think about looking at the repurposing of existing structures with minimal changes in the way fold reconstruction people often do, and then looking at these as sort of attractors to the viable states that tell you where a spandrel can be anchored, to make a kind of comprehensive evolutionary reconstruction that is both what you do and also makes contact with the strong parts of what other fold people do?
Okay, so we need to do some groundwork here, right? So what we're looking at right now, these are structural modules, whether they are functional modules or not, are not necessarily discrete pieces of sequence. And we need to get to that state in order to do things like ancestral reconstruction methods, to see if we can figure out what do the intermediates between two types of modules on the span would be. And I'm very much inspired by the work of Brian and Orban at the University of Maryland. I don't know if you've seen some of this work, where they essentially go from a 3-helix protein that binds to BSA to a 1-helix 3-beta sheet protein that binds to an immunoglobulin. And what they're able to do is, through a series of single amino acid mutations, keeping the binding sites for both of those domains intact, walk you from a protein that has one structure to a protein that has another structure. And at the very center of that, with a single amino acid mutation, you can go to one structure or you can go to the other, right? And I think that what we're saying here with spatial connection being an evolutionary connection, that is, I think, at this point still a hypothesis. And the only way for us to really see whether that's plausible is to go into the laboratory and try and design some of these pathways and see whether they're plausible. So the way that I approach that as a protein engineer will be to try and actually engineer some of these transition fossils and see whether we can make them behave the way that we would expect them to.
It was a really intriguing talk. Thank you so much. So in terms of like the transition from this prebiotic to the biotic function, one of the key question is the maintenance and the evolution of the functionality of this polypeptide that co-associated with the metal. And I was wondering, so this type of minimal module could form in prebiotic era. However, we all know that the protein can be only being replicated through this genetic coding, and that's always been problematic. But I was wondering, it seems like this minimal module have almost minimal sequence specificity, meaning that it doesn't necessarily need to be this specific sequence in a primary mode. So have you ever looked into like the phase space, the functional landscape of this type of module, and if that landscape is big enough to basically cover a wide range of different combination of amino acids that can actually do this redox cycle, then do you think that will leverage the error catastrophe that was thought to be necessary for this genetic code problem?
So what you're asking, if I understand and clarify me, is that did we come across the one 12 amino acid sequence that works? Or is this
Is
just part
like
of a very
exactly.
evolvable sequence? And the answer is that we've only tried 2 or 3 sequences, right? And we have one that doesn't work, and we have 2 that do. But those are they are not a very good. If we were to answer the question that you're asking, we wouldn't design the sequences. We would build libraries,
Right.
and then we would see what is the success rate for that. And I think that's a very good thing to try, definitely.
Especially like given it's the backbone, it seems to be the key, which doesn't require the side chain. Could it kind of imply that?
That's right. I mean, all of the interactions here are either cysteine. The cysteines have to be there, the 4 cysteines, and then everything else is backbone. So in theory, this should be highly designable, highly evolvable, but we haven't shown that yet. Yeah. I can't talk to you until you get the cube.
Thanks. Yeah. Nope. Yeah, thanks for the great talk. I have 2 questions. One's maybe easy and the other one's a little bit harder. I think in 2008, Leslie Dutton's group, another piece of work out of that group, they created these things that they were calling the maquettes. And I think they had 16 amino acids. And I was just wondering what's the commonality or difference between what you're showing and what they were showing?
So that sequence is very similar to this. If you look closely at this, you can essentially see that there's a cysteine, 2 amino acids, and then another cysteine. The difference there is that they have 3 amino acids between, and they're glycine, so they're very flexible, right? So there's not necessarily a specific conformation for those. And what I would argue, the reason why I think this peptide is so well behaved in terms of both yield, specificity for iron-sulfur binding, and then also redox stability, is that it adopts, we've essentially addressed the Leventhal paradox for this. It has a unique conformation in the apostate that we believe is entactic, is pre-organized to bind the iron-sulfur cluster. And so in that sense, it's better behaved. But in a lot of ways, it's similar, right? Essentially, what they did also was to go into ferredoxin, see what was the spacing between cysteines, and then just make a model peptide, a maquette, that had that same spacing. And essentially, all we've done here is, rather than look at the sequence, we've looked at the structure, tried to figure out what are the key elements of the structure that give you that iron-sulfur binding, and make a maquette that mimics that.
Okay, and then the second question that
that was the easy question.
Yeah. So maybe a little bit speculative, but for these 4 iron 4 sulfur cubes, the possibility of site differentiation of the cluster is really important for like a conatase activity like Joseph talked about, but also the entire radical SAM protein family has a similar mode of binding for, in that case, acetylmethionine. So the site differentiation, having this open iron coordination, seems important. Do you think it will be possible to make an open coordination iron
We're
site?
definitely going to try, right? I mean, we're definitely going to try. We're going to try variants where we remove one of the ligands, or we replace them with imidazoles. Try different combinations of things. So, if for example having a 3 iron 4 sulfur site is advantageous, or whether we can stabilize that 4th metal with hydroxyl that can then be replaced with an active site ligand, then that would be very exciting.
I think so too. Yeah.
With peptide, with P loop, it wouldn't matter at all. Would it?
Pardon me?
With a P loop, it wouldn't matter at all.
With a P loop. Well, I'm sorry, I don't understand. What would matter?
Phosphate loop.
Yes.
And you've got a structure like this, about 7, 6 or 5, as long as 2 or 3 glycines in there, or it's heterochiral.
That's right. So have the glycines in there so that you could access that left-handed conformation, right, rather than using non-natural amino acids. Yes.
So you could say that's right from the very beginning. You probably got a P loop right at the beginning, very early in life, and all you've got is P loops everywhere.
P loops, yeah. So and this shares a lot of similarity to P loops. I mean, essentially, in a P loop, all you have is a row of amides pointing at your nucleotide. So electrostatically, this behaves a lot like the P loop. I think what's unique about this typology is it also presents the primary shell ligands in a side chain conformation so that they can hold the metal covalently.
Yes, so just one question about that then. Well, by the way, are there glycines in the 12 amino acid ones?
No, there's no glycines in this. So it's all
Is it
it's
heterochiral?
L and D amino acids in this.
Sorry, they're all the amino acids.
They're L and D amino acids.
Could
so
you
it's all-
imagine a loop like that without cysteine? Could you imagine
Oh,
sequestering
absolutely,
it?
absolutely. Yeah, you could, but then all the interactions would be electrostatic, and so maybe then that could bind a phosphate, much like these cationic nests do, or other anions.
I ask because it looks though cysteine is quite hard to make early on in life.
So let me ask you this. So Mike, what were the thiol-based amino acids before cysteine? There must have been a thiol.
Oh, there were thiols, but they probably weren't amino acids.
So what were they that were, I mean, we can make something structurally, anything we want. We don't have to use the natural alphabet here. What would you suggest is a possibility?
Well, you mean something like an ornithine serum?
Okay. I mean, we just need to make some bonds between amino acids to make a ring or a linear structure, a maquette, if you will. But the point is, we understand, of course, that imidazoles and thiols and the cysteines are not found in chondritic meteorites. So what would you? But there were hydrogen sulfides.
I mean, Bianchi published.
Yeah.
40 years ago.
So what do you give us? What do we
give
get to
us?
play with?
What do you want?
Just glycine.
Oh, no, well, actually, I prefer alanine and arginine. My favorites are alanine, especially, and asparagine, and
asparagine, okay.
And probably aspartate.
Okay.
And I think I'm kind of stuck with those three to
Okay,
start with.
so we like aspartate, maybe not for iron sulfur, but for binding other types of metal clusters, definitely. So like manganese oxides, for example.
Anyone have a last question for the speaker? In that case, let's
Thank you, everyone.
thank Vic once again.